Okay, in this video, we're going to look at the cross-bridge cycle. Uh, you should have read about the sliding filament model of muscle contraction. This is the accepted mechanism of how muscles contract. And it says that during muscle contraction, the thick filament slides the thin filament towards the M line of the sarcomere. That causes the sarcomeres to get shorter. If sarcomeres are getting shorter, well, myofibrils are getting shorter. And if myofibrils are getting shorter, the muscle's getting shorter. The muscle cell's getting shorter, which leads to the entire muscle getting shorter. So the cross-bridge cycle shows us how the thick filament causes sliding of the thin filaments. And we break it up into 4 steps. Before we get into the 4 steps, let's first consider that in order for the cross-bridge cycle to occur, in order for myosin to interact with actin, the myosin binding site on the actin molecules, the little black spot there on each of these actins, has to be uncovered. When a muscle is relaxed, normally this goldish thread, which represents tropomyosin, is blocking that binding site. In order to get it to become unblocked, what has to happen is the sarcoplasmic reticula has to release calcium ions within the muscle cell. Those calcium ions then bind to troponin, the yellow spherical molecule that we see behind the calcium ions. When calcium binds to troponin, troponin shifts, and that pulls tropomyosin away from actin's myosin binding site. So before this can happen, all that other stuff that I just kind of narrated has to happen. So let's imagine all that other stuff did happen. Now the cross-bridge cycle can happen. Let's take a look at it. So it is a cycle. That means once we get through the last step, 4 in this case, we come back to step 1. And we could really start at any of these 4 steps and it would make sense. And that's another aspect to it being a cycle. We're going to start here. Our textbook calls this cross-bridge formation. So what they're talking about is the myosin heads are able to reach out and bind to actin molecules, and this forms a temporary cross bridge between the thick filament and the thin filament. In order to reach out and bind, myosin has to be energized, and the energy driving the cross bridge formation has come from the hydrolysis of ATP, which has happened previously in step 4. Notice that we see at the ATP binding site, we see ADP and phosphate. Those are the byproducts of breaking down ATP. You get ADP, phosphate, and energy. And that energy, at least some of it, was used to form the cross bridge. Okay, so cross bridge is formed. Let's move on to step 2, which is called the power stroke or the working stroke. So ATP has been hydrolyzed. That provided the energy for the cross bridge to form. Well, myosin still has some energy left over from that ATP hydrolysis. And what it's going to do with that is it's now going to pivot. So we see this little arrow here indicating that the myosin head has pivoted. Remember, there's a hinge region associated with each myosin molecule, and the heads pivot at that hinge region. When the head pivots, that slides the thin filament towards the M line of the sarcomere. Like one unit, whatever those units are, we just got 1 unit closer to the M line of the sarcomere. Now, one thing to keep in mind, we're looking at the cross-bridge cycle happening on the right side of the sarcomere. The same thing is happening on the left side of the sarcomere, but in the opposite direction. So we're seeing a thin filament on the right-hand side of the sarcomere being slid towards the M line towards the left. The same thing is happening over here, except the thin filaments are being slid to the right towards the M line. Okay. After the myosin head has pivoted, ADP and phosphate are released from the ATP binding site, and that allows step 3 to happen. Step 3, cross-bridge detachment, a new molecule of ATP comes along and binds to the ATP binding site at the base of the myosin head, and that causes the myosin head to release from actin. Without the new ATP binding, this releasing can't happen. So the cross bridge is detached. Now we're going to see in step 4, we're going to hydrolyze this new molecule of ATP. The textbook calls this cocking of the myosin head. We're getting ready for it to do its thing again. So that new molecule of ATP has been hydrolyzed. Now the myosin head is re-energized and ready to go back to step 1. And this can happen over and over and over again to produce the amount of muscle contraction that we're asking any given muscle to do. Okay. Now, when it's time for the muscle to relax, the calcium ions are going to be removed from troponin and pumped back to the sarcoplasmic reticula. And once the calcium ions are removed, troponin shifts back to its normal position, which causes tropomyosin to also shift back to where it's blocking the myosin binding site on the actin molecules. All right, so that's a wrap on this one.